Hi, in this video I would like to describe a sagittal section through the head of the fetus obtained from an extrauterine pregnancy outside the uterus. Extrauterine pregnancy is a life-threatening condition. Basically, the embryo and later the fetus has no chance to develop and survive in the environment outside the uterus, and it's a great threat to the mother's life as well. The fetus is of anatomical age 8+4. That means 8 weeks and 4 days, which is already after the embryonic period, so we are talking about a fetus, not an embryo. The crown rump length of such very young fetus is about 27 to 31 millimeters, and its weight does not exceed 3 grams. In terms of gestational age, which means the time from the onset of the last menstrual bleeding, it would be a pregnancy of 10+4. That means 2 weeks more than the anatomical age. We can divide bones of the head and pharyngeal area into 2 groups. The neurocranium, which contains the bones providing support and protection to the brain and sensory organs, and the viscerocranium, comprising bones of the face and pharyngeal arches. Bones anywhere in the body can be formed by 2 mechanisms: endochondral ossification and intramembranous ossification. The term endochondral ossification basically means creation of bone on a cartilaginous base. The chondrocranium represents the base of the skull, from which we see mainly the emerging sphenoidal bone and part of the occipital bone, which are formed by cartilage. The term intramembranous ossification represents creation of bone directly from mesenchyme. Most skull bones, as well as the lower jaw, are formed by intramembranous ossification. The mesenchyme for the skull bone formation originates either from paraaxial mesoderm or from neural crest. The border between these 2 sources in the base is the hypophyseal fossa, but the situation is probably much more complex. Let's have a look at how the facial area is formed first, before we get to the developing brain, which takes up most of the slide. In humans, 5 pairs of pharyngeal arches are formed along the sides of the foregut. Pharyngeal arches are covered by ectoderm on their external surface and endoderm on the internal surface. They are filled with mesenchyme, which is derived from the mesoderm of the lateral plate and the paraaxial mesoderm, or predominantly in the head area from the neural crest. Mesenchyme derived from the neural crest is called ectomesenchyme. In the slide, we can distinguish oral and nasal cavities, which are already divided by the palate. The palate is a horizontal plate made up of bone and soft tissues. We can see the formation of bone in our slide. There is a tongue visible inside the oral cavity. Posterior 1/3 of the tongue develops from the hypopharyngeal eminence, derived from 3rd and 4th pharyngeal arches. Anterior 2/3 develop from 1st and 2nd pharyngeal arches as 4 swellings, which fuse together. This corresponds to the innervation, which is divided between the trigeminal nerve and the facial nerve in the anterior 2/3 of the tongue, and the glossopharyngeal nerve and the vagus nerve in the posterior 1/3, with the motor innervation of the hypoglossal nerve, as the muscles of the tongue are derived predominantly from occipital somites, migrating ventrally. Okay, this was kind of complicated, but I hope you get it. In the center of the tongue, we can see the developing skeletal muscles in the form of myotubes. The bones of the 1st to 4th pharyngeal arches are from the ectomesenchyme. The basis of the 1st arch is the transient Meckel's cartilage, from which the malleus and incus in the tympanic cavity are formed by endochondral ossification. On the contrary, the mandible is formed by intermembranous ossification in the area surrounding the original Meckel's cartilage. Stapes, the third middle ear ossicle, develops from the cartilage of the second pharyngeal arch, called Reichert's cartilage, which also forms the upper part of the body of the hyoid bone, and stylohyoid ligament is also built from the mesenchyme around. In addition to the developing tongue, we can also see the primordia of the teeth in the dental cup stage inside the oral cavity. The dental cup is formed by the invagination of the surface ectoderm of the dental lamina, which is divided into a layer of outer and inner enamel epithelium. There is a reticular epithelium between these layers, and this is one of the two places in the human body, apart from the thymus, where we can also find reticular epithelium. A layer of ameloblasts is formed from the inner enamel epithelium. Ameloblasts produce the enamel. On the contrary, odontoblasts producing dentin are formed from the surrounding ectomesenchyme, which thickens in the area of the dental cup, forming a dental papilla with odontoblasts on its surface. Thus, the tooth is formed from two sources: the superficial ectoderm of the oral cavity and the ectomesenchyme from the neural crest. In the area of the sphenoidal bone, we can see the developing pituitary gland in the area of the sella turcica. The anterior lobe of the pituitary gland, or the adenohypophysis, is composed of three parts: pars distalis, pars tuberalis, and pars intermedia. It arises as an outpouching of the ectoderm of the primitive oral cavity called the stomodium, in the form of a so-called adenohypophyseal or Radke's pouch. The pars distalis and pars tuberalis arises from the anterior part of the Radke's pouch, and the pars intermedia is its posterior part. There is a cavity in between that can give rise to remnants called Radke's cysts, which are a common finding in the pituitary gland in adults. The posterior lobe of the pituitary gland, or the neurohypophysis, represents the nervous tissue. It originates as an outgrowth of the diencephalon and remains attached to it by the infundibulum. To sum it up, the pituitary gland has two origins, which fuse together to make up a small but crucial endocrine organ. And now it's time to stop ignoring the largest structure, which occupies most of the slide, the developing brain. The central nervous system arises from the neural tube that is derived from the neuroectoderm. The dilation of the neural plate produces three primary brain vesicles: rhombencephalon, hindbrain, mesencephalon, midbrain, and prosencephalon, the forebrain. During the fifth week, the primary brain vesicles form 5 secondary vesicles, which can be observed in our slide. Myelencephalon, from which the medulla oblongata is formed, metencephalon, giving rise to the pons and cerebellum, mesencephalon, midbrain, diencephalon, future thalamus and hypothalamus, and telencephalon, endbrain. These brain vesicles may look different in different slides. This is just a section, and since there are cavities within the brain, we can see various sections of these cavities as well. There is the fourth ventricle between the brainstem and cerebellum. Next, we have a part of the third ventricle within the diencephalon and the parts of the lateral ventricles within the telencephalon. The cranial portion of the neural tube undergoes flexion. We distinguish 3 flexures. 2 appear earlier: cephalic in the mesencephalon, cervical between the myelencephalon and the spinal cord, which is not visible in our slide, and one later, the pontine flexure between myelencephalon and metencephalon. The cerebellum originates from the rhombencephalon by joining of the so-called rhombic lips. They create a cerebellar plate, which we can observe here. There is also the choroid plexus, a structure producing cerebrospinal fluid. The area where the rostral neuropore has closed is represented by the terminal lamina, lamina terminalis. Cell differentiation begins in the rhombencephalon area, where the arrangement of the layers is identical to that in the spinal cord. There is the ventricular zone adjacent to the central lumen, from which all neurons and glial cells, well, except microglial cells, differentiate. Microglial cells are of mesenchymal origin and reach the central nervous system later as monocytes. The cells from the ventricular zone migrate into the appropriate layers and form an intermediate or mental zone, from which the gray matter develops, and marginal zone, from which the white matter develops. In the area of the telencephalon, where cerebral cortex is formed, the situation is a bit more complicated, and several layers appear here, which do not correspond to the zones in the spinal cord and the brainstem. In the layers of the cortex, it's again essential to know that cells, whether glial cells or neurons, developed from the ventricular layer that surrounds the central cavity, in this case the lateral ventricle. From this area, the cells migrate, leaving only ependymal cells that line the ventricular system. This process creates several layers that give rise to gray matter, or cortex, and white matter of the brain. In our slide, we can distinguish the aforementioned ventricular zone, which is still quite cellular at this stage. The first neurons produced from the ventricular zone form a superficial layer, the preplate, which underlies the developing pia mater. Axons extend from these neurons and form the intermediate zone, future white matter. Other neurons migrate into the preplate and split it into the marginal zone, future molecular layer, and the cortical plate. Early neurons of the cortical plate will form lamina 6 and 5, while later born cells will form more superficial laminae 2, 3, and 4. The migration of the neurons in the gray matter is important for the proper development of the brain, and migration disorders can lead to serious developmental defects. We can appreciate the olfactory bulb bulging from the developing telencephalon. The emerging structures of the eye can also be seen on some slides. Optic vesicles evaginate from the forebrain and come close to the surface ectoderm. The lens arises from the lens placoat, thickening of the surface ectoderm and its subsequent invagination. Optic vesicle will develop into the optic cup, and we can distinguish inner layer of the optic cup that will form neural retina. But I don't think that we can see that on our slide, or I guess we can see this pigmented epithelium all around. It's not very good section. Normally, there is an intraretinal space existing between these layers, which is not completely closed in the area of the optic part of the retina. Thanks for watching. This video is a brief overview of the development of the head area, and certainly does not represent a complete picture of its development. If you would like to know more about individual parts and their development, let us know in the comment section below.